And good morning, everyone. All right, so the test is set for Monday morning, and it is not designed to be a 2-hour test. I'm pretty much building it like I would build the test for in class, but you'll have 2 hours, and I hate Moodle, so this thing is going to be placed in the Moodle as an assignment, which means it'll be just a Word document, the exam, and you can print it out and turn it in. You could do it completely electronically and repost it, whatever. Just make sure you put your name on it. And yeah, so I thought what we'd do is kind of go back over some of the sort of lists and things and comparisons we've made, and review from that perspective a bit. It's sort of one of the ways I think about this is organizing it in kind of a chart fashion and thinking about the story of what we're talking about and keeping up with it that way. Let's see how that goes. So, we start out talking about the structure of the atomic nucleus as a way of introducing the idea of bonds, and specifically as a way of discussing covalent bonds and then the weak-ass bonds, ionic and hydrogen bonds, and polar, nonpolar interaction stuff, because that plays a critical role when we start thinking about protein structure and the way enzymes work. So we then turn our attention to the basic macromolecules. I figure, sure, we'll throw that out, because this is a sort of an obvious question of the monomers and the polymers that they make in terms of biochemistry, and what are some of the interesting bits about these individual polymers. So, just to start, our biomolecule polymer chart begins with my favorite and yours, the carbohydrates, and of course the monomer that makes these guys up are sugars, are also monosaccharides, and you then generate polysaccharides, sugar polymers, and a couple of examples and things we talked about. And we'll be throwing the idea of polysaccharides around from here on out. These are the sugar polymers. Their main claim to fame is going to be they all have a pretty strong negative charge, and we have the ability to form lots of hydrogen bonds. One of the things I was thinking about, as I imagine on the exam previous form, I would ask how you draw two water molecules engaged in hydrogen bonding. And remember, this is the fact that oxygen, being electronegative, has got a slight negative charge, and the hydrogens each have a slight positive charge. And so we make this charge interaction that works. And it allows water to be very sticky. Water is both adhesive and cohesive, as we say. It also is giving water a significantly higher boiling point than what would be predicted. And just thinking about water made me thirsty. All right, so the other part of this is that polysaccharides have a tremendous ability to hold water, and we also have their ability to function as thickeners. So the idea that we go from a long chain carbohydrate that has internal hydrogen bonds, that is within the molecule or intramolecular, and then when we heat it, it becomes they're being raucous. We have the intramolecular hydrogen bonds. You heat it up, and the molecule opens up, and then as it cools, we begin to form intermolecular hydrogen bonds. And this creates a gel-like material. So it's how you make Jell-O, but it's also actually a part of the thickness and gooiness of various fluids lubricating joints, and it provides for a lot of interaction between proteins that have a lot of sugar on them, glycoproteins that are often on the cell surface, and later in the semester we'll be talking about proteoglycans that are mostly sugar with a little protein core, and they're maintaining the extracellular matrix. They provide spots for cells to bind, hold a lot of water. They're squishy, they're good lubricants. Another part of the carbohydrate story is the standard sort of glucose polymers, cellulose and amylose. Cellulose being wood, amylose being starch, and this underscores the idea that enzymatic processes are very much bond specific, because we do not have cellulase, but we do have amylase. We can digest starch, great, but we can't touch wood. So, there you go. And, well, that's what I have to say about sugars. Mainly hydrogen bonding, hold water, and it's, of course, a good energy source as well. All right, the next bit in our chart then would be the fats and or lipids class, and the monomer polymer model kind of falls apart a little bit here, but we've got as the monomer more or less the fatty acid, and the fatty acid can have it has the carboxylic acid group along with a bunch of carbons, typically 18, 16 or 18. There just are a limited number of fatty acid scaffolds in living systems. And the ability for these guys to have their melting point determined by both the length and the presence or absence of double bonds. And so we can have a trans fat in which we've got the, kind of a small fatty acid, but regardless, you have the hydrogens on opposite sides, and these are not typically naturally occurring. We can typically see the cis structure for our fatty acid, and the presence of double bonds is going to lower the melting point. So double bonds mean that the fatty acid with its kink can't pack together as tightly, and so it's a bit more liquid and less, its melting point has been lowered, and so consequently it's going to be liquid and fluid at colder temperatures. The next step in the fatty acid story would be making a basic polymer of the mono, di, or triglyceride. So glycerin is the 3-carbon scaffold that we're going to link the fatty acids to. And it's one of these things where having a little bit of structure is not necessarily a dangerous thing, actually a good thing, because we can think about how we're going to link these guys up, knowing that we're going to make a molecule of water. In the condensation reaction, we've got the carboxylic acid tail here. And what we do is pop an OH group from one side and an H from the other, and make a molecule of water. And so we condense out in a condensation reaction a molecule of water, and we make a link. So we have a monoglyceride. We can have a diglyceride. We can have a triglyceride, which is how fat is typically stored. But more importantly, we could add here a phosphate group. And just because I have multicolors, I'm going to draw it in a different color. So we can add, oh, I like that red, the phosphate group. And we know whenever you hear the word phosphate, you were immediately thinking massive negative charge. And this then becomes a phospholipid. The standard phospholipid, in addition to having the phosphate, also has a decorating group, very often a choline molecule. And this is a nitrogen-containing big heterocyclic molecule. You can also have amino acids attached to it. You can also have sugars attached to it. Mainly, you've got this big charged head over here. And so we can then roll this phosphate up, and we end up with the amphipathic molecule that we diagram thusly, where we've got the polar head and the nonpolar tail. And when we mix these guys together, hilarity ensues. We end up in water forming first a my cell, not your cell. And then if you stir this up, you add a little bit of salt, you can actually begin to form the lipid bilayer, the phospholipid bilayer. Because the cytoplasm is aqueous, the external environment is aqueous, so by having the polar heads facing to the outside or the cytoplasm, we have a system that works. And we'll be getting into membrane transport in a little bit, and this plays a key role there. Let's see, other things with the fatty acid lipid class. We would be negligent if we did not mention the fact that in here we can use an amphipathic molecule as a detergent. Because phospholipids allow us to form an emulsion. They act as emulsifiers in that you've got the fatty acid tails able to interact with fat molecules or nonpolar molecules, and the phosphate head is able to interact with water. So we've got the micelle now. If we start adding just straight fatty acids or nonpolar molecules, grease and oil, they can get trapped in here and then wash them down the drain. So we have detergents. Detergents are very good at disrupting membranes. That's why you're encouraged to wash your hands. Viruses are coated in a phospholipid bilayer. You wash your hands for 30 seconds, but not 29 seconds. And you will then begin breaking apart the membrane surrounding the virus. And without a membrane, it is just a piece of nucleic acid, very easily degraded, not going to be infectious. Wash your hands. So, that's the fat class. Just to throw in here, because if I didn't mention it here, we wouldn't mention it anywhere. And that is steroids are put into this class as well because they're nonpolar. Steroids, typically based on cholesterol, which I will not bother to draw, and you should not bother to memorize. Please do not. Steroids are a class of heterocyclic molecules. They get turned into different hormones. Steroids also, like cholesterol, can fit into a phospholipid bilayer and actually act as a stiffening agent for the phospholipid bilayer. So that's the fat class. And I'm thinking, anything there I forgot? Now, move on. So from here, we turn our attention to the last two polymers, proteins and nucleic acids, and we got a bit more to say about them. So the next class then in our list are going to be proteins. These are, can you read that? Not as well. Sure, I'll go back to my fat marker. Proteins. These are amino acid polymers. So the monomer is the amino acid, and we've got 20 distinct side chains or R groups that we can point to. This is another one of those molecules where if you're going to start taking biochemistry in the fall, over the summer, make your R group flashcards and start memorizing them now. But for us, no, we don't need to memorize them. But knowing the structure, the base structure, is useful for thinking about how we can link things together. We've got the amine group. Let's see how the markers are drying out. The amine group with 2 or 3 hydrogens, depending on the pH, and a central carbon, and then a carboxylic acid N. So we've got a hydrogen here, and then this is where the R group is attached. The simplest R group is a hydrogen, and that makes the amino acid glycine. But these 20 different R groups, 20 different amino acids, they can be categorized, they can be polar, or consider them to be polar, non-polar, they can be acidic, they can be basic, they can have aromatic rings, all kinds of stuff. All right, so just as another little term, when we talk about a molecule being acidic, it typically means it has a net negative charge. All right, and basic is a positive charge. So from here, we can begin linking together amino acids to form amino acid polymers, i.e. proteins. And one of the things that a typical question would be, diagram and draw 2 amino acids linked in a peptide bond, point to the carboxy terminus, point to the amino terminus. I may draw it out for you and ask you to label them, but if we were to link these guys together, let's see, yeah, we'll do it over this side. I hate linking it from this side because you always have the first amino acid is line leader, and this amino terminus stays where it is, and nobody messes with it. But this back end is where we add new amino acids. So we've got an amine function group, this is the carbon, carboxylic acid, R2. And again, we're going to make a condensation reaction. So we're going to pull off an OH and a hydrogen, and bada bing, you have made the carbon-nitrogen bond. It's just a covalent bond, but it is given the specialized name. It is the peptide bond. Now, because we've been talking about transcription and translation, we can actually elaborate on this specific point. The peptide bond is generated by the enzymatic activity intrinsic in the large subunit of the ribosome, known as peptidyltransferase. When back in the 60s, guys were figuring out how amino acids were linked, one of the things that was being looked for was, what's the enzyme that catalyzes the formation of the peptide bond? And it turns out it is in the large subunit of the ribosomal RNA. It's just one of the proteins there. It also takes a molecule of ATP that is hydrolyzed to drive this reaction. It's one of the reasons why we say the production of amino acid polymers is a fairly energy intensive process. You got to make the individual amino acids. You also have to burn an ATP to make each bond. And so you start thinking about a big amino acid polymer with a couple of hundred, 1,000 amino acids. That's a pretty large energy investment. One other thought that we can throw in here is that we can link through the carboxylic acid to the 3' end hydroxyl of the tRNA. Let's see. Go upside down. So we've got the OH group here because we know that tRNAs are RNAs. They've got both hydroxyl groups here. I'm doing this upside down. And here is the base, and here is the phosphodiester backbone, linking it to the rest of the tRNA. And if you're getting confused by this, don't worry about it. Part of what I'm thinking is that by having a base understanding of some of the structures, when you're asked or you're thinking about, how is an amino acid coupled to the tRNA? Well, you know, it's got to be coupled this way. And you have the free amino end. And so this is displacing this amino acid from its tRNA, and we're just going to keep on building. So, things to think about. All right, so we've made a peptide bond. Now, the really critical and interesting point with amino acids is the amino acid polymer now, because of these 20 different R groups, gives us the opportunity to build a molecule that doesn't have just straight same charge homogeneity like a sugar molecule does. I mean, it's just negative charge. And you can make great jelly, but it doesn't really form up into interesting structures. Now that we have a protein that has got the 20 different R groups, we have the possibility of these multiple layers of structure, which of course are the primary structure, which we define as the amino acid sequence, and it's held together by the very tough covalent bond. We have secondary structure, which is local R group interactions. So who are the R groups on either side of you? Oh, probably 5 R groups on either side of you. And now we're going to have all the weak ass bonds. We're going to have ionic, polar, nonpolar interactions. Hydrogen bonds. Things that we can very easily manipulate and break. The third level of structure is now folding this guy up. These are now non-nearby, non-local R group interactions. And they're using the exact same weak-ass bonds. So, from this falls out the fact that A, proteins are pretty darn delicate. B, we can have really minor changes in conditions that will begin disrupting secondary and tertiary structure. You're going to have some really serious, nasty conditions to disrupt primary structure. There are enzymes that do it, proteases, but short of that, you're going to have to throw it into very strong acid or very strong base or raise the temperature up to probably 450 degrees in order to break the primary structure. But secondary structure, tertiary structure, weak-ass bonds, they will begin to change if you talk harshly to them. If you stare at them and give them a mean glare, the hydrogen bonds break. Secondary and tertiary structure are very delicate. And so changes in temperature begin to unfold the protein as we raise it up. As we lower the temperature, there are like non-standard hydrogen bonds that might be able to form because this thing's not flexing so much. We change the pH. We can begin to take weak acid R groups and actually make them neutrally charged rather than negatively charged. And even if we begin to drop the pH low enough, they will become positively charged, push things out. You can take an amino acid that is marginally soluble in water and start adding salt to it. And that's going to cause this marginally soluble guy to say, I can't bear it anymore, and fall out of solution. All of these kinds of secondary and tertiary disruptions, if they are disrupted, we say that the protein has been denatured. So we can determine primary amino acid sequence by looking at the genome. Secondary and tertiary, you have to use computational analysis, and you can come up with a good idea, but you don't necessarily have the exact secondary and tertiary structure. We do have quaternary structure, but as I always say, I refuse to put that up there. Quaternary structure is when we have two different proteins linked together or interacting together. But because you can't determine that from the genome, I refuse to talk about it, and I didn't say that. All right, so we can go on and on. One of the things we can do with proteins is we can add sugar molecules to produce what we know as a glycoprotein. We can have a kinase, an enzyme that adds a phosphate. A kinase will add a phosphate, big negative charge, group to a neutral amino acid. And that will dramatically change the shape of the protein structure. There was, like in the sample questions, I don't think I've asked this question in a long time, but the idea of having two proteins, one from the pig, one from the cow, whatever, and there was a neutral amino acid in one of the guys that was changed to an aspartic acid in the other guy, and that change makes the protein slightly active all the time, whereas the guy that has the neutral amino acid requires a kinase. And that actually is something we do see either evolutionarily or a classic way of generating a tumor is to have a phosphate or a negatively charged amino acid substituted for a neutral amino acid in just the right spot. All right. So that, what this does is provides the basis for pretty much everything we've been talking about. We could go on and on here, but we got ground to cover. Let's continue on with our fourth biopolymer. And it's a little bit of a chicken and an egg kind of phenomenon of which one is the most critical. Proteins are nucleic acids. We have evidence that life might have initially began as a nucleic acid-based kinds of biochemistry, but very quickly you get into amino acid-based biochemistry, which really allows you to do all kinds of different song and dance. So the next class is the nucleic acid class, and the monomers are the nucleotides, and polymers are RNA and DNA. Nucleotide, the thing you need to know is the 5-carbon sugar, the ribose sugar. We've got the 1', the 2' hydroxyl, 3' hydroxyl, 4' carbon, 5' carbon, and here we've got the 3 phosphates, the triphosphate. We use this hydroxyl, so just 1', 2'. This is the guy that we're going to link the next nucleotide to in the phosphodiester backbone. This is the OH group that you use to decide if you are a ribonucleic acid, or if you remove it, you are a deoxyribonucleic acid. So it's whether you're an RNA or DNA molecule. Let's see. Oh, is there anything else to say? So we start adding phosphate links here and creating the nucleotide chain. Nucleic acids have the ability to interact through base pairing, where the adenine base pairs with guanine, the thymine base pairs. Oh, geez. I'm screwing that up. I was thinking about two different things. Sorry. Adenine and guanine are purines. Man, that was kind of odd. Adenine base pairs with thymine. Guanine base pairs with 3 hydrogen bonds with cytosine. All right. So this is important or interesting because when we start thinking about things like the Tata box, the TA rich region, that's what we also generically refer to as the -10 sequence in the promoter. It is AT rich because that's the spot where RNA polymerase unzips the double-stranded molecule and begins reading the bottom strand. So the CAT box, as it's called, or the GC -35 spot, that's the binding spot for RNA polymerase. But it makes sense to have an AT-rich region for where you're going to open it up, because you're only having to disrupt 2 hydrogen bonds per base pair rather than 3 hydrogen bonds per base pair. So another way of kind of thinking about it from a mechanistic perspective, you know they have the CAT box and the Tata box. Which one is where? The Tata box is the -10 site where you're going to start opening up. The CAT box is where you're going to begin binding. All right, so nucleic acids. And the terminal phosphate of the adenosine triphosphate ribonucleotide is often the phosphate we think of in terms of energy transfer. What you're doing is taking that really big negative charge and putting it somewhere. So that chart covers a ton of ground. Another important part to our general discussion, and it really kind of falls out from this, is when we start getting into metabolism. And we'll be getting into some of the basics of metabolic pathways in a little bit. But for now, we can really just think about the role of enzymes and how they're operating in terms of carrying out biochemical reactions. So we went through some of the basics of thermodynamics, where we have this idea of a spontaneous reaction, which, even though the name is spontaneous, it doesn't mean it happens spontaneously. It just means that it is thermodynamically favorable. And spontaneous reactions are what make up the catabolic type metabolism processes. And then we have, from here, a net energy release. And we'll be getting into this in a good bit more detail when we get into metabolic pathways, but it's good here. And then we've got the thermodynamically non-favorable reactions, we refer to as the non-spontaneous reactions. And these are making up the anabolic side of metabolism, and these are energy requiring. And a great way to put energy into a system is to add a phosphate and cause a protein to dramatically and very quickly change shape. Slap it together, pull it apart, whatever. All right, so this leads us then to how we can carry out biochemical reactions. And we were looking at primarily these spontaneous catabolic types as a good model place to start. And we can think about a good kind of catabolic reaction with our energy graph here, where we got molecules with high energy being glucose and O2. And then we got molecules with low energy being our good fire putter outers, water and CO2. So energy is being released as we are having a chemical reaction occur. So we can have the reaction progress, the reaction direction, this way, and we have all this energy being released. Now, of course, blessedly, we're not at risk of having sugar packets spontaneously explode. That's a good thing. It is a spontaneous reaction, but sugar is not a particularly dangerous kind of compound you can have in your kitchen. And that's because we do have this speed bump we got to get over, or an energy bump, and that is, of course, known as, you see that? No, you can't see that. Uh-oh. See, I'm thinking about you viewers at home. Yeah. All right, so there's energy speed bump. This is what we know as the energy of activation, or the activation energy. And the way we're able to carry out biochemical reactions, of course, is by lowering the energy of activation, making it something that doesn't require 400 degrees, but only 37 degrees. And this, of course, is how we could define an enzyme. An enzyme is a protein, typically protein, catalyst. And now we have to define catalyst. A catalyst lowers the energy of activation. Yay. All right. So we can have enzyme catalyzed reactions. And that leads us then finally to looking at reactions and things that are being carried out by enzymes. So a standard way of looking at enzyme catalyzed reactions is to have some kind of enzyme assay that we can determine a rate. And then we can look at the enzyme rate in comparison to the concentration of the substrate. That is the stuff the enzyme's acting on. And a critical thing to keep in mind is that enzyme catalyzed reactions are going to have a typical logarithmic kind of curve in which we have at low substrate concentration, a logarithmic increase in the rate. And then we get to a spot where we have plateaued out. And this is because the enzymes are physically interacting with the substrate. And with a given number of enzymes, there is going to be a given rate at which they can break down the substrate. Now, you could jack with this by adding more enzyme, but that's not how you do the experiment. So we then have this idea of this point, this rate, being the maximum velocity, or Vmax. And we can then make a comparison at 1/2 Vmax. We have a substrate concentration that is given the special term of the Michaelis constant, or Km. And what this is a way of thinking about the relative affinity a enzyme has for its substrate. But actually, we can think about this with any kind of interaction. The relative affinity that a protein has for a nucleic acid sequence, just as a way of thinking about this. So when we start talking about the RNA polymerase in prokaryotes, their Km for DNA in the absence of sigma factor is kind of high. They exhibit this rather loose binding, a non-specific binding. When sigma factor is bound, all of a sudden the RNA polymerase has a very high affinity for the promoter sequence, and that's the Km changing. So Km is actually a really cool way of thinking about molecule interaction. The lower the Km, the greater the binding affinity, and the greater the affinity interaction is. And oh my God, I see people writing that down thinking, I hope that's right to say. It may be a bit of a generalized statement. If you take advanced enzymatics as a graduate student and I'm wrong, hey, I'm saying it now on record. I might be wrong. Except now, lower Km means greater binding affinity. All right. We then threw in some ideas about different kinds of inhibitors. We can have a competitive inhibitor. And a competitive inhibitor is some kind of molecule, drug, but some kind of molecule that is primarily going to be mimicking the substrate. And what's going to happen is Vmax is achieved, but at a higher substrate concentration. So, by definition, because Vmax is achieved, but it's going to take more substrate to get there, we can generate a curve that'll look like that. So, you did make Vmax, but because of that, the Km is further this way on the x-axis. Km is larger. The alternative to a competitive inhibitor is, of course, the non-competitive inhibitor. And a non-competitive inhibitor is essentially taking individual enzymes and pulling them out of the game. In that we have effectively lowered the number of enzymes. Again, this is sort of my way of thinking about it. And a non-competitive inhibitor is blocking the ability to achieve Vmax. So Vmax is never achieved. And one way of thinking about this, at least in my model, is because you're not actually going to be affecting the enzyme's ability to interact with its substrate, you're simply taking some of the enzymes out of play. Those remaining enzymes, they're still chewing the way they always do. And consequently, if we have something like that, the non-competitive inhibitor never achieves Vmax, And that means that we have a new Vmax at 1/2 Vmax. Well, if you draw it out right, and if you do the enzyme calculations right, generally what happens is Km is not affected. So, yeah. All right. So we can think about enzyme inhibitors this way. And there you go. All right. So last, but most recent, and this is the chapter I gave you all the figures for, and you got the book too, the whole idea of transcription translation, but we've been covering that for the past few days. So let's just think about a few specific points. We've got transcription. This is the world of the RNA polymerase. You have to have the promoter. And in the eukaryotic system, we've got 3 RNA polymerases, Pol 1, Pol 2, Pol 3. Pretty much all we've been paying attention to, and all I ever want to talk about is Pol 2. In prokaryotes, there's a single RNA polymerase. So we've got, in both cases, the upstream promoter sequence and regulatory regions. We've got +1, which is the first nucleotide of the RNA. And then we've got the structural gene, that is, that which is being transcribed. And so we have an RNA molecule coming out. Now, a key point in eukaryotes is the role of RNA processing. And the list of RNA processing events is the addition to the 5' cap, which we know to be critical for protein translation initiation. We've got the 3' poly A tail, which is critical for RNA stability. And we have, most importantly, exon intron splicing. So, last then point would be, in the last minute, the translation differences between prokaryotes and eukaryotes is primarily dependent on the translation initiation, in that the prokaryotes, they're going to have the small ribosomal subunit binds to the ribosome binding site, aka also known as the Shine-Dalgarno sequence. And a very cool experiment that's been done in bacteria is you can change the nucleotide sequence of a Shine-Dalgarno site. And then you can make the complementary change in a portion of the ribosomal RNA, and everybody's working just fine. So we know that the ribosomal RNA in the small subunit actually is base pairing with the Shine-Dalgarno sequence. And that sets up where AUG is and where the open reading frame is. The fallout of this is we can have the ability to read and generate multiple open reading frames we can have multiple regions of one RNA being individually translated. When we look at the eukaryotes, initiation is happening at the 5' cap. We then undergo what is known as the scanning model, in that rather than having a ribosome binding site that you land on, you just start crawling along the RNA until you find the initiating AUG. The AUG in the appropriate context of the Kozak sequence. That's the AUG that you start up at, and you go. So the fallout of this is a prokaryotic RNA has the possibility of generating multiple proteins. A eukaryotic RNA, by definition, once it's been processed and out in the cytoplasm, is a single protein that can be generated from it. That's all I have to say. So, a good way to kind of begin working with this is abstract your notes, start writing your own charts like this, and condensing it. That's what you got to do, and get it into a story that you can kind of link together. Because now that we've been talking about some stuff, there's a lot of threads you can start pulling together on. Cool. That's all I know. We won't meet on Monday, but I'll see you on Wednesday.